So I want to start by welcoming you all. Usually, and I hope everyone is well. It's been a very unusual year, so I hope everyone's doing well and looking forward to the winter break. This is our building in Canterbury, the Ingram building, this lovely yellow fronted building on a quite nice sunny day. And this is where a lot of our goes on. I have an office in there. This is where our forensic labs are. This is where we do our teaching usually. And I want to start by giving you a couple of slides to introduce you to our course and talk about kind of what is the core of forensic science at Kent and what it means to us. And it's embedded in those three things across the top there of employment, enjoyment, and engagement are the key things for us. We're trying to inspire you and help you realize your potential. And from that perspective, I apologize. I'm just going to go out of this for a second because there we go. That's better. From that perspective, there we go again. From that perspective, I want to introduce you to our core forensic team. And these are the 7 of us you see on the screen there. So this is our core forensic team. We, of course, because we're part of the School of Physical Science, we're actually part of a much larger group of lecturers from across our school and also working with lots of other schools from the law school, from biology, from anthropology. A whole set of people work with us as well, but this is our core forensic team. So along the top there, we've got Donna and Chris. They're our directors and deputy directors of undergraduate studies. We've got Stuart Gibson here. He's our admissions officer. So if you have any admissions questions, or as you're going through and applying with us, he's the person who'll be looking past your applications. We've got our newest member that's just joined us is Andrew Langley here. He's joined us from the Met Police in London, and he brings the true police practitioner experience with him. And alongside that is not Dr. Bob, but is Bob Green, who's also a former practitioner in forensic science, is also our director of student engagement. He spends a lot of time worrying about and spending time making sure that we communicate well with our students, make sure we engage you all the way through our process. Dr. Aaron Burko down the bottom here, he's a lecturer in chemistry and forensic science, and he works a lot with you in your first couple of years on our course. And myself in the middle here, I'm not only am I a lecturer on forensics and chemistry here, I'm also the director of public engagement and communications across the school, which means I'm responsible for how we communicate with you from all our audiences through from when we talk to you as students, when we welcome you at open days and taster sessions like this, all the way through to when we're trying to find you a job at the end of your course, how we engage and how we communicate with employers, industry. So our core forensic team called the 3 E's, employment and engagement. Employment being, we place employment in the middle of everything that we do at Kent, because at the end of the day, you're going to be going out and wanting to find a job at the end of this. So actually, we want to embed this from the start and all the way through. We use our fantastic links with various employers from all over the country, in fact, all over the world. We work with them to make sure that we are teaching our students to be the best and most employable when they go out there into the world and look for it. Enjoy it. It's important that you enjoy it. We have a great community within SPS, within our school, and we're in the forensic science team. All of us get on fantastically well. We have lots of social events going on. We have a fantastic engagement, which is our 3rd E, with our Forensic Science Society, and engaging with you through doing lots of formal, informal activities. Every year we have a big trip through the society going somewhere. So we've been to a variety of places in the past. We've been to Interpol previously out in Lyon. Previous years, we regularly go up and engage with, go to high courts, go and work with forensic practitioners and invite forensic practitioners from other countries over to talk to us. And in fact, just this week, we were awarded from the National Coordinating Centre for Public Engagement, we became the first academic school level, so subjects level institute in the country to be awarded a silver engagement watermark for all of the fantastic work that we do in engagement across everything that we do. And we embed all of this, and within our training and our 3 E's, we also have our crime scene house, which is a bespoke facility, allows us to really various crime scenes. And as your years of your course, you'll start from learning about how to apply the techniques and how to kind of find particular bits of evidence at a crime scene. Through to stages of the course, you'll actually be in there. We will have staged a crime scene. We will have staged various scenes, and you will go in as part of a SOCO, a senior crime officer, actually going trying to look and see what evidence you can collect from a particular scene. And we have this a big area with the house and the gardens, a resource we've invested alongside all our fantastic chemistry and physics labs that we have within our main building. So that's a very brief kind of introduction to our course and what we do and what we believe in. I want to go on to then spending a little bit of time talking to you about of mine and give you kind of a bit of a taste picture in science at Kent. This brings together elements of 3 or different courses you'll be taking across your time at Kent. And a particular thing that we do, which is really as you go towards your latter years, your 3rd year and your 4th year, if you're on your master's course, you actually also spend some time working on a project work, doing a bespoke project to do with forensic science. And that's when you work side by side with our fantastic researchers, Kent, to actually try and do real research, which will have real impact in the real world. So I want to take you on this kind of journey through some of the techniques and touch on a few forensic things that and some of the potential jobs you can have, and then take that through to some real research that is going on by our postgraduate students and some of our undergraduate students in their final year of their course at Kent right now, and see how you'll be able to go on this journey if you join us at Kent. So I want to start with the question, what does a forensic scientist do? A forensic scientist, when you say a forensic scientist, you probably start by thinking that it's something like this. We've probably got, we're probably thinking of, some kind of TV forensic scientist, maybe CSI, maybe NCIS, those kind of things are what we're thinking about as a forensic scientist. Maybe we're thinking about someone like Sherlock Holmes. There's another real early example of applied forensics, and those are the kind of things that we talk about when we do science. And we could have a long conversation about whether or not that's realistic or not. In fact, we have people doing final year projects. So these project modules I'm talking about who are doing work. Their project is in fact taking TV forensic science and seeing whether it would actually be real and it would actually exist in the real life. So an example of one of those projects we've taken is whether you could 3D print bullets, or whether you could 3D print a key from just a single impression in order to be able to open a lock. Sort of things that in forensic science and you see on TV, but seeing if that's really true in the real world. So that's what TV tells us forensic science does, and a lot of it is quite good. Some of it's really awful, but this is what my perspective of what you as forensic scientists will do together, particularly at Kent. So Kent, and what forensic scientists do, we take aspects of physics, we take aspects of chemistry, we take aspects of biology, and we take aspects of law, and we merge all of those together into a single thing in order to bring you and round you up together to be a forensic scientist. So at Kent, we don't have those clear divisions. You at school, maybe you would have learned some physics, some chemistry, some biology, maybe you've considered some law or something like that. And on top of that, there's lots of other things. There's forensic psychology and lots of other things. But at Kent, we don't make that division. We bring all of those together, and we bring all of those different subjects together in order to be focused on forensic science and what you as a forensic scientist can do. And one of the reasons we do that, it comes back to our 3 E's and the first E, which was the employment. That's because in the real world, there aren't these clear divisions. We all work together to try and solve problems. So we don't, in the real world, I used to work in industry. So I used to work in the food industry, and in the food industry, I came into it with a particular set of expertise, but I would work with people with a huge variety of different expertise, all working together in order to try and solve problems In what I was doing, I was working in the food industry, but for forensic scientists, the same. We work and we bring aspects of physics, chemistry, biology, law, engineering, psychology together in order to address forensic problems and in order to gather evidence and bring convictions in various ways. And that's exciting because you will become so rounded as a forensic scientist by the end of your course, because you'll bring all of these expertise together. And it can really embed your expertise, embed your kind of knowledge in all of these and the core science in your expertise so that you come out the other side of it. So I want to highlight 3 potential roles that our students go on to do after they leave Kent. The first one is, I referred to it earlier, a SOCO, what we call a SOCO, which is a scene of crime officer. These are the people you may be familiar with, those ones in kind of these noddy suits, the sort of thing you come over here. In the middle, we have analytical chemists, and so analytical chemists from all over, going from working in big, we have some students who've gone to go and work in big kind of biochemical labs. We have one student who's been working very closely with one of the big companies that's rolling out a vaccine for COVID-19. In fact, currently they've just got a job there. So working on some of the analytical and the quality control problems and interests that you can have there. And Fingermark Development is a bespoke part, often linked to the crime labs, but even Fingermark Development is another example of a tool and a thing that we study as forensic scientists. So I want to start discussing Fingermark Development a little bit and looking at that role and what it can be done and what it would mean in the real world. So when we think about Fingermark Development, we think about something like this. We think about a forensic scientist, a scene of crime officer at the scene of a crime, in a car, in wherever it is, at a house, wherever the crime might have taken place, trying to identify where there might have been finger marks left behind, and then collecting them, gathering them for their use, potential use for a conviction or linking someone to a crime in the future. And that is the core of what Fingermark Development is. It's one of the staples that has been around forever within forensic science, of being able to try and identify a human and link them to a particular crime. And I want to do that, but I want to actually, so in my lectures, usually I try and bring some interactive elements into what we've done, and I'm going to try and do this with you guys at home, so hopefully this will work. I want you to go to, someone's already done it, so it's on the top of the slide there, but I've also put it into the chat for you now. If you go to pollev.com/Dr. Rob, then hopefully, there you go, pollev.com/Dr. Rob, and then hopefully you'll be able to see a screen where you can answer this question. Fantastic. And on the screen, there'll be 2 options for you. It will either say, does this work? So I essentially want you to say, yes, you can see it. Or if you want to be a bit difficult, please go to say, I managed to respond. What more do you want from me, Rob? So please feel free to do that. Give you a couple of seconds. There we go. Fantastic. There we go. Oh, which one's winning? Oh, brilliant. Okay, so thank you. I can see people are in there and are responding to this for me now. Thank you very much. So this is one of the tools that we like to use when we're teaching to be interactive. We can do this in a live class. We can also do it online. So now we've done that. My next question for you is, what is a fingerprint? So is it dirt, fat, water? Is it all of those things or something else? So I'll give you a few seconds to vote. Fantastic. Oh, people change their minds. Fat, dirt, water, something else. Brilliant. Very good. People changing their minds. I like the fact that people are changing their minds. That's all right. I'm going to actually, what I'm going to do is we will before people change their minds anymore, I'm going to actually lock that poll just so we've got it there. So we can see. So actually, most people said that it was you thought it was all of the above. A few people said it was just fat, and 9% of those who are in this lecture thought that it was something else. What I can say is, for all of you who said all of the above, you are absolutely correct. In fact, a fingerprint is all of those things and lots more things. But the core parts of what make up a fingerprint and a finger mark when it's left behind is it is some dirt. So it is some material over here, like our dirt that we have here. So it is some dirt. There is plenty of water in there because there's water from the sweat that we've left behind, that whoever the perpetrator or the person has left behind when they left their finger mark. There's all the oils from the stickiness from your hands. Those are all also in there, all mixed in with dirt and anything else. Dirt could be anything. As we'll come to later, dirt could also be some drugs that a perpetrator may have picked up previously or something they've picked up and left behind. It could be anything else, any other material from their surrounding. So in fact, it's a lot of other things, but in fact, a finger mark is a combination of all of these and many, many more. And how does a fingerprint actually absorb to a surface? I want to take you through, think about the lifetime of a fingerprint and how it absorbs to a surface and how that evolves over time. And initially, when you handle anything, when you pick up a bottle, when you pick up a cup, when a perpetrator handles a knife or handles something else, or someone handles some drugs or some notes, initially that finger mark is left behind. If it was on a porous surface, everything, that fat, that water, the dirt, all of those things sit in a nice kind of blob on top of the thing. And this is our finger mark sitting on top. Immediately after it sits out there, a few minutes to hours afterwards, depending on how porous the surface is, they start to sink in. So particularly the water-soluble components, so the water, maybe some of the fats, if they are water-soluble, start to sink into our porous surface, and they sit across here, and they sit across the bottom, whereas the water-insoluble components, that's our other components there, maybe the dirt and the other bits that might be residue left behind sit on top. And then eventually, after many weeks and months, if you come to a scene of a crime much later, or you're gathering evidence, or if maybe it's a case that is being readdressed and you're trying to gather more evidence from some evidence that's been taken from a crime scene, and you're re-looking at it on that surface, then eventually you end up with an absorption. So most of the material, the salts, the urea, all those water-soluble components are sitting in here, in a corona around this, and they're fully absorbed into there. There's also going to be all the proteins, all the amino acids, all the other things that you might have left behind sitting in a layer in between. And then finally, on top, you have all your water insoluble components. You have all of maybe your dirts that weren't soluble. You may have any other residues that might have gone in there which haven't made it into there. So that's the progress when we think about it. And that's important for us because when you're approaching a scene of a crime, you don't know. Maybe it was a fresh finger mark that's been left behind, or maybe it's something that's been around a while. So the timeline of how long something is taken there and the surface and the properties of the surface are all important factors that you have to take into account when you're analyzing and trying to pick up a finger mark. So my question for you is, which part of a fingerprint do most fingerprint techniques target? And you should have an image on the screen in front of you now if you go back to polev.com/Dr. Rob, where you were before. And you should have an image there in front of you. And I want you should be able to drop a pin. You can see one of your colleagues has already done this, and say which part does most of the fingerprint techniques target. Fantastic. We can see those votes coming in here. People changing their minds. Looking at it. Fantastic. We've got a variety of different votes for different parts of the finger mark there. Some people are saying that it's in the kind of the water soluble bit that we have down the bottom. Some people are saying that it's further up in the thing. I'm gonna tell you, sorry, I'll go back to that briefly. In fact, people are still voting. I'm gonna lock the votes now just so that we can see and talk about it. There we go. Thank you again for everyone for voting on that, for those who are doing it. I like to, in my lectures, I like to have lots of interactive elements. So that's kind of an important part of what we try and do, to see some feedback in an anonymous way. So you have an opportunity to see how you're learning and build that into your learning as you're going along. So actually the answer is most finger mark or fingerprinting techniques, so things for picking up finger marks, target the very outer layer, the insoluble stuff on the surface. Maybe some of them also target the proteins underneath, but most of it is the residue of the very top layer, the layer that's left on behind on top of everything. It's that very outer section that you can see on the screen here. It's those very top ridges are the key thing that most fingermark techniques look like. And that's what we see. You can see on the image on the left-hand side there. That's the material that's come from maybe the fatty deposits. That's maybe some of the other residue that's left behind. So it's great. And that's what, if you look at some of the fingermark, kind of the fingermark detection powders, the fingerprinting powders that you'd be used to, applied with, for example, a brush. And there's a variety of different brushes that you can use and you can pick depending on the material you're looking at, on the surface you're looking at, and various other things. Most of them are sticking to that very top layer, that residue, that's what's left behind on top of everything. And then you take that, as I said, and you can pick different powders depending on what material you're looking at. I've put 3 examples here with their chemical structures underneath, and anhydrine, the various fluorinated versions, and DMAC are 3 examples of these. And we use those chemicals, and you as a forensic scientist will learn when to use and when to pick each of those chemicals, one of those particular powders or other chemicals, to be able to identify and visualize a mark, depends on the material you're looking at. And part of the importance when you're looking at a finger mark is not only being able to detect it, but also to be able to then identify it and visualize it in some way. So all 3 of these are good examples of kind of cutting edge techniques, and all of those 3 of those are targeting particularly the amino acids. So that bit I was talking about, so the bit that's maybe absorbed and maybe the insoluble stuff that's sitting on top. So those 2 layers. So anyone who said either of those, that's the material you're looking at. A lot of the most current techniques focus on the amino acids. So that was our middle layer in that thing, as we were talking about, because even if you've had some kind of weathering, some kind of aging, maybe some kind of washing of a surface, you're still able to pick up some kind, identify some part of this from the amino acids. And those are particularly good and easy to identify using fingermark detection techniques like these. However, we have a problem. This is the cutting edge of science. You will learn this in your 1st and 2nd year. These are the kind of techniques that you will learn during your 1st and 2nd year. Less than 10% of all marks collected at crime scenes in the UK are actually used. In fact, I think on things, the percentage is somewhere around 7 or 8%. So that's a tiny number of finger marks that we've collected that are actually usable in court. So despite the fact that we've got all this research and we've seen the crime officers have spent a lot of time identifying and finding these marks, collecting the evidence and then identifying these marks, in fact, such a tiny amount is usable in court. And a lot of the reason for that is because you aren't able to pick out a lot of the structure. So you can identify a mark, and even from a partial mark, you should be able to get enough identifying factors to be able to identify if to link that to a particular person. But the problem is that might not be strong enough, because all you're getting is linking to that residue, to that amino acid layer. So it's affected by potential smudging or wiping, because cleaning products would easily resolve that. It's affected by the sensitivity of the technique itself. It's affected by even being able to truly link this, because of even a small smudge will pick this up and lose it. So what we said, and this is research that we do in my team, I'm a researcher, I've worked with the police across the UK to say, well, this is a problem. How can we change this on its head? And one thing we said is, well, obviously everything at the moment is, as we said, targeting those amino acids or maybe the insoluble component. But what if instead of picking those bits, the amino acids or the insoluble components, what if instead of doing that, we go for the holes in between? So instead of trying to find where the finger mark is, so finding all those ridges from the ridges on your finger that you'll be able to see, instead we find the holes where there isn't anything. We make a negative, as it were, of the finger mark. Now, why is that interesting? And that's interesting because of the fact that instead you're not relying, most of those techniques we've talked to now are very kind of, there's either a yes or a no. You either the chemical is going to stick to the finger mark, stick to those amino acids or the other components, or it's not going to stick. So when you get your finger mark, you just see your line and you just trace it. But for those of you who might be interested, already know about forensic science, you actually know that that's just the primary structure. So that's just the most basic information that you can pick from a finger mark. In fact, if you look at your fingers or look at your fingerprints or finger marks, those marks have a curvature, have a shape, have a lot more texture to them, which can't be picked up with these techniques. But if we were able to use a technique to sensitively choose between picking it as looking at it, so not just sticking to the amino acids, but also looking at the gaps in between and picking everything else in between with a resistive response to it, then you might actually be able to pick a lot more information. So we did this. We worked with a couple of police forces in the UK, and we worked with CAST, who were the centre a lot of these techniques in the UK. And I think this was the Mags and mine, Rob Bucker. We did this and we did the opposite. So we essentially targeted the gaps in between. And particularly we used it for targeting the gaps on metal objects and to start off with. So starting up, looking at kind of the gaps on a knife or another metal object, scissors or something else that a perpetrator might have used, a gun, more so in the US than in the UK, but still the same. And we were able to use this to actually identify much more high resolution pictures of a fingerprint by, instead of targeting where the finger mark was, by targeting where it wasn't. And from that negative, we could get much more information about the finger mark. To the point that we found from our initial trials, and I say this is now being used by police forces and a number of forces across the UK, this is 2 to 3 times better than current techniques. So we've gone from where about 7 to 10% of finger marks are usable and admissible in court. We've now moved up towards the area where 20 to 30% are actually capable of being able to make a link, potentially lead to a conviction if you can make a link between those things. So that's exciting because suddenly we're able to advance it using research that we have developed at Kent, and there have been students from our forensic science students who've actually been involved in the development of those things that has gone out into the real world and is really affecting the usage and used by senior crime officers and fingermark experts in the UK at the moment. In fact, we spread out and it's starting to be used in other countries as well. So that's the first thing I wanted to talk about. And in fact, you can get even more from a fingerprint. And this is something that we learn at Kent. You can get even more from a fingerprint because you can start to use a technique which is called mass spectrometry, which is this very complex bit of instrumentation that I've shown on the screen here. And it's part of a course that we teach because we embed chemistry and analytical tools so heavily within our course at Kent, and we make sure that you're trained to be able to understand these. So what mass spectrometry is a very complex thing which fires electrons at surfaces in order to get information out of it. And what it does give you is we can even use it to get spatial molecular information. So we're able to not only find out in the finger mark, we could identify there is or isn't a finger mark, but we could also be able to then map that finger mark in order to identify what particular chemical species were within that finger mark. And you can do this by taking out these very complex instrumentation. A mass spectrometry instrument 10, 15 years ago took the size of a room, and you can make it much smaller and make it to focus onto a single point. So this was used in the food industry. Here on the left in the food industry, they use this. In my prior work, if you were interested in what was going on the surface of your tomato or whatever else, in fact, what you would do is here, you were in fact firing a stream of electrons at the surface of that tomato, very light, and then chipping off materials, and you're actually able to use that and collecting what is chipped off and actually using that to measure the molecular species that are in there. What if you could do the same with a finger mark? Now, obviously, this is actually here for demonstration. You wouldn't actually put your finger in this beam because that would be dangerous over time. But the idea that if we're able to get map this molecular information on a tomato and across the surface of the tomato to see if there's contamination or something else going on, you can do exactly the same on a finger mark. And that has been done. And this was done in order to, and here's an article here which is now quite old, it's 10, 12 years old, where they were able to actually map finger marks. To actually take particular points across a finger mark and identify what the chemical species were in there. And in fact, in this case, it was able to even identify that not only was there a finger mark, but there were particular drugs, substances of abuse that were embedded within that finger mark. And that was able to be used not only to identify someone and link someone to a position, but also give even more high resolution information to identify that that person had also been in contact with an illicit drug at some point in that process. Which, remember, our jobs as forensic scientists is to be able to bring together information in order to potentially link people to a crime or bring a conviction in court, or provide some evidence in order to lead to a conviction in court. So this is providing even more evidence, or even more information. And everything I've talked about here, if we go back to our initial starting point, everything I've talked about here has required using knowledge of chemistry to analyze a biological system, and in fact, using physics and engineering in order to go even more high resolution in what you're studying, and all of that within the context of law, in order to be able to lead it for conviction, because that is always where we want to sit in the end. So as a forensic scientist, you build all of these expertise and all of these skills in order to help us learn and understand what we're doing. And you will learn that, and we learn this as we go through our course at Kent. So I've alluded to this a little bit. So if we go back to our second point of potential jobs, we started to talk about analytical chemistry jobs. As I said, I used to work in the food industry before I came to Kent. And there I used to work a lot with, we were interested in quality control. We were interested in, making sure that there wasn't any bad contaminants or anything else that was going on there. Exactly those things are interesting in forensic science. In forensic science, we want to be able to identify. If we've taken a suspicious substance from a scene of a crime, we want to be able to identify exactly what that is, why that's there, why is it interesting. So we want to be able to identify if we take some kind of red substance from the scene of a crime. We want to identify not only getting DNA evidence, but we might want to identify if there's any other components of that blood or that are of interest. Maybe there's some other stain at the scene of a crime. These days, at the moment, you have simple presumptive tests that allow us to say yes or no, that particular stain at the scene of a crime is potentially some kind of bodily fluid. Is it yes or no? Maybe you're starting to identify it. What if we could get much more high resolution information about it, as I was talked about? Now, the problem is, in order to get those high resolution information at the moment, we have to take everything we've got and take it back to a lab. And that's what happens in the forensic labs. They have fantastic, this is an HPLC machine that you can see on the slide here. We are able to take this and gather this information in a lab after it's been gathered at the scene of crime, but that takes days. And this is so often where forensics on TV breaks down a little bit, because of the fact that in real life, that isn't actually what happens, and it's not that simple and easy to gather information so quickly. It takes days in order to do this with very meticulous processes. And that's important to get the legal thing. But the trouble is, because the kit is so big and bulky and heavy and hard to use and needs those experts in order to use it, you need to have all those training, and it needs to have a big infrastructure in a lab to do it. So the speed isn't there. We're not able, in the case of, for example, a capping or something like that, when you might want to have a very rapid turnaround in understanding in order to respond to the situation, we don't have the speed to be able to do that. We're not able to respond quickly. We're not able to bring the portable things together. So I asked the question, because this is one of my interests, and this is what we do. What if we can take this analytical kit back into the field? So what if we can take these big bulky bits of equipment, shrink them down so that they can then be taken back into the field where evidence gathering teams, where our senior crime officers are, in order to give much more rapid response to any investigating officer, so they can actually say, oh, and provide more information in order to respond rapidly to a particular crime. And we do this. I work on this a lot within the group, and we're working on exactly this, trying to take these analytical techniques and kind of reducing them down in size, so that you go from something that's large to something that sits in the palm of your hand. And I have a number of people, a number of students who work in my team. I have a number of students who have worked in my team previously, who've graduated and then stayed on to carry on doing this research with me, working on these techniques. And one of the primary techniques that we use is a technique called microfluidics. And microfluidics works by using the flow of liquid and its interaction with surfaces in order to separate particular materials from it. And the best analogy I want to take for that is I want to take you to the analogy of two pieces of paper. Both of these things, you'll probably have them at home. You'll probably know them very well. You have a piece of paper like this, kind of normal writing piece of paper. You have a piece of paper like we have on the left there, which is kind of a normal material that we would use to blow our nose. And I want you to think about this. I'll do this at home. I want you to think about which one, how they would work and which one would work best. What would happen if we dropped those two materials from the sky? What would happen and how would we see it? And in fact, for this, I'm going to take a second and actually I'm going to stop my sharing so that you can see me for a second. And there we go. So imagine you've got that. We've got our two materials here. We've got our piece of paper on the right here, and we've got our tissue paper on the left. And I want you to think about this, and I'm going to get up for this demo, if you can see me. Okay. And think about what happens. So I start with these two here. And if I drop them, I want you to think about this at home. Which one's going to hit the ground first? You think it's a piece of paper or the tissue? You can try this at home if you want. So I'm going to drop them. You won't be able to see this, but actually the tissue paper. So this one hit the ground first. Our tissue paper that we blew on nose hit the ground first. In theory, both of them would probably hit the ground at about the same time in a normal thing. And how did we know that? We knew that because of the fact that we know about the properties of these things. These are both paper, they're both paper-based materials. We know that because this one's floppier and it deforms much more, it's going to change and it's going to maybe catch the air and deform and hit the ground first. Now I want to take the same idea and imagine that this piece of paper is interacting with a surface. And for that, I'm just going to rip off some pieces of paper. And I also want to use this because this is a festive lecture. I want to put on my festive jumper to help me with this. So got my festive jumper here. The students who know me and who've seen me and I lecture on this course know that around Christmas I like to rock a nice festive jumper. And so I've got my friend here. I've got Rudolph and his nice red nose, three-dimensional nose here on the surface. And I want to come back to our two pieces of paper here again. And I've dropped off our two pieces of paper, and I'll bring it forward a bit. And now I've just ripped off a little bit of them. So we've got our tissue paper on the left and our writing paper on the right. And I want to ask the same question. What's now going to happen if instead of dropping these pieces of paper on the ground, I drop them on Rudolph's nose? What's going to happen? How are they going to interact? I'll start with the piece of paper on the right. So let's have a look. We dropped the piece of paper. It hit Rudolph's nose, kind of sat there for a bit, but then fell off very quickly, in fact. Let's begin with the tissue paper. The tissue paper has dropped and sat on, hit Rudolph's nose, but in fact, it's still sticking there. And it's still not moving off. And you can see that there on the camera. And that is for exactly the same reason. We knew this. In fact, I'm sure a number of you at home were able to predict this already. That was that was going to happen. The tissue paper was going to stick on Rudolph's nose, whereas the writing paper was going to fall off. And we know this instinctively because we know the material properties. We know the material. You can't see my wonderful jumper properly, as if you went in 3 dimensions, but you know that that's going to be a soft material. And we know that the paper is more likely to just fall off it, whereas the tissue paper is more likely to stick to it. That's what we know because we know those material properties. What if we took that knowledge of material properties and scaled it down to the molecular scale, to the size of a few molecules? What if we could use that same knowledge of knowing how hard or soft or how few molecules are going to interact on the surface? We'd actually be able to use that same knowledge in order to separate and design and identify particular elements for molecule. And if I have to share my screen again, there we go. Fantastic. that is exactly what we do, exactly that same approach, but with a few molecules, and that allows us to be able to identify particular things. And we do that through a thing called microfluidics. So I'm not sure how well this will work at home, but we use by using microfluidics, where in fact you will see that some of the red blood cells, if you follow them, the red material, so this is what blood looks like. We've got the white blood cells going through. They're the white blobs that we have on screen. We've got yellow blobs, which are the platelets that we find in blood. And the red blobs, these are our red blood cells. And what we've got in here is a series of pillars. You can see them all over. There's a pillar here. We have all of these pillars. And if you follow it, for example, let's look at some of these red blood cells. You'll see the white blood cells, they just flow down as we flow liquid passes, as do the platelets. But some of the red blood cells, actually, as they start to interact with the surface, here's an example of one red blood cell here. As that starts to interact with the surface, it actually starts to go in one direction. It interacts with the surface just in the same way that our tissue paper interacted with Rudolph's nose, and then starts to stick on it and then maybe move in a different direction. So that's the same instance that we've just seen with paper on Rudolph's nose, but done with blood in a microfluidic device. And if we can take that knowledge, we can actually create what I like to call a racetrack on a molecular scale for different components of blood, where if the red blood cells are always going to hit those pillars and move in one direction, while the white and the platelets move in a different direction, over time, we're going to separate the red blood cells from the rest of the blood cells. And so we're able to identify a particular component from a very complex thing like blood. We're able to separate and then identify a particular component there. And we can do this, not just in that example for red and white blood cells, but for much more complex mixtures, potentially mixtures of any substance that we identify at a crime scene. Instead of having to have a large bit of infrastructure in order to identify it, we can go down to scanning something. That on the screen, incidentally, that you can see on the screen is one of the things that's not real blood. That's just, in this example, that's just some food coloring. But this is, for example, if you think of like an iPod case or something like that, that is about the size of an iPod case. So it fits in the palm of my hand. Able to do exactly the same thing that previously we would require an entire room in order to analyze. So by doing that, we're starting to be able to put things back into the hands of scientists in the field at the point where they require it, away from having to have a separate analytical lab in order to study it. So that's how taking us on the journey through identifying and looking at a particular materials, how we've changed things for Fingermark development through analytical chemists, from the kind of core things that you'll learn in your first couple of years at Kent on our course, through to what you will learn and you'll have the opportunity to work on, working in my team or my colleagues at Kent, to really change these for the future. The final one I want to talk about is how else we could change the job of a senior crime officer. And this is an interesting question. And for me, this is a thing that I've recently got interested in, maybe in the last 3-4 years, thinking the biggest problem with a senior crime officer is the fact that the officer has to go into the scene of the crime in order to identify anything. So that could lead to contamination, which has a huge effect. That could lead to the officer themselves being contaminated. A classic example of this was in the Salisbury poisonings. In fact, it was the police officer who went into the house of the Scripples, who became one of the most affected by, or because he was in hospital for so long, because he entered the scene of crime and he actually handled the material. He handled the doorknob where it was. He then brushed his face and rubbed his eyes. And that was what actually caused him to get infected and take this in. Now, obviously, because he wasn't expecting it to be a crime scene, he wasn't wearing full protective kit like these people in the photograph are. But still, that problem exists, that currently senior crime officers enter a scene often without any knowledge of exactly what's going in there and without able to prepare and potentially contaminating or causing other issues. So I asked the question, what if we could replace a senior crime officer, a SOCO, with a drone? And we're doing this. We're working, and this is quite an old photo of an old drone that we had, in fact, it's an old stock photo, by actually looking at putting instrumentation onto drones in order to fly them as a first port of call into a crime scene. So we can get rapid, real-time, non-intrusive access to crime scenes. You no longer have someone walking in or touching surfaces. You can have something which is much more controlled, actually passing through a crime scene and identifying the particular things that we're interested in. And we do this by not only going in and having visible cameras on there, we do this by putting a whole variety of different sensors onto the drone in order to be able to identify what's in there. And that comes back to what I've talked about before, because we care about being able to look at these things, but a drone is reliant on, has a battery life. And if we want to put heavy bits of kit on there, that battery life isn't going to last very long. So we have a problem. So it comes back to before about actually wanting to reduce and miniaturize these things a bit in order to study it. And we do this for a variety of different things in order to study and gather information. One of the primary ones for another project that we work on with, in fact, I currently have one project student, so one third-year student working, one final year student doing his, in fact, master's, because he was on our fourth year forensics course, actually working on this now with my PhD student in my group. And we're actually interested in doing this, not only for looking at crime scenes, but also for looking at cows. So here it is. In fact, here is a drone on a farm. And we built, if you can see on the screen, we started to build from Raspberry Pis and various other things, essentially a sensor set that we can strap to the bottom of the drone that's lightweight and low powered, but can then be used to focus and gather information. And we were we were interested in thermal information. We were interested in finding out things for the interest of cows. We were interested in finding out whether or not a cow was sick, because this is the same idea of shrinking technology down, like your senior crime officer is interested. But we also know go back to thinking about, why do we care about cows being sick? We care about them because if there's an outbreak of a disease on a farm, suddenly the farmer doesn't want to cull all of those cows. He wants to be able to identify very quickly. In fact, we've got a very relevant thing for now, where if any of you have been over the summer, went through any airports during the pandemic, you would have found that you walked past a load of thermal cameras, which would have been used to try and identify whether people had an elevated temperature. Now we're trying to do exactly the same thing, but with cows, which comes back to my favorite question. Where is the best place to take? Now, I haven't got a quiz for this, but I do want you at home to discuss with your parents or your friends, whoever you've got, where's the best place to take a temperature on the cow? Now, I wonder how many of you are saying this, because this is well known that the best place to take a temperature on a cow is its bum, because that's what we see. That's where vets always seem to take a temperature. In fact, I mean, I don't really know why vets do that. That's not the best place to take the temperature of a cow. Well, I do know why they do it, but actually there's a lot of error in this. That is not the best place to take a temperature. In fact, the best place to take a temperature on a cow is in its eye. In fact, here's a thermal image of a cow. And you can see the kind of more white the image is, the hotter it is. You can see around the eye, in fact, specifically in the corner of the eye, is one of the hottest places on the cow. And that's the best place to take a temperature reliably and repeatedly on a cow. So in order to do this, we've taken all of our techniques, we've taken physics, we've taken chemistry, we've looked at biology and then an engineering thing, and we had to bring in our last tool in order to do this, which is computing. So we're working with computing experts in our department. We've got Dr. Stuart Gibson, who was our admissions officer I mentioned at the start. He is also, he designs and works on a lot of machine learning and computational diagnostics for forensic uses, as well as being involved, and he has his own spin-out company, which was used for the analysis of for EFIT software that a lot of our police forces use across the country. And he's a computing expert. So working with him and some colleagues, we're actually looking at doing something I thought I would never say, which is facial recognition for cows. So that when we have a cow, not only can we identify the cow and identify its face, but we can also identify the features of the cow's face and look at the corner of a cow's eye and use that to actually specifically identify that, and then take a temperature from it from a thermal perspective. And here it is. In fact, I've got an image on screen here. Here is an image of a drone, one of our drones with those cameras on. In fact, it's an image of our drones, one of those drones, filmed from another drone, because why not? Why don't we have one drone and then film it from another drone? And there it is with all of those on in a field in Scotland. In fact, this was on a farm in Scotland. now let's look at what that drone looks like. This is the thermal camera on that drone. And you can see, in fact, you can see in the background there, you can see there's some cows, there's very hot objects. You can see us waving there, and you can see identify the different features of our heat maps on us. So we've actually developed a technology that can be deployed on a drone for identifying lightweight imaging technology, which before, incidentally, would take up the size of a room, to be able to start deploying it with the right wavelengths on a surface. So that's the last, and I apologize, I've just seen that my internet has been bad. I'm sorry if you've had any issues about that. I hope the recording should have worked okay, and hopefully I'm happy to talk about any of these things that you may have missed in the questions afterwards. So, in fact, this is my last slide, and this is the last thing I wanted to say. This is the journey that we go through as forensic scientists and doing our forensic science at Kent. And I'm going to stop there and just talk directly to you. As forensic scientists at Kent, we go through all of those things. We start from understanding the fundamentals of what is currently used in the world of forensic science, of analytical sciences, of all the other things we do. And then we take it all the way through in order to look at what the future is and analyze future, developing in our labs in the final year, developing kind of final techniques that can be used to resolve and change the world of the future, so that future senior crimes look and ways they're analyzed look very different from the way it is today. I want to leave it there, and I want to say, firstly, apologies if there were any, the quality was bad at any point in there. And I want to introduce Luke, who hopefully is there. Introduce yourself and say hello, Luke. There you go.
Hi, I'm Luke. I'm second year forensics student. Yeah.
Perfect. No, yeah, that's perfect. I apologise. I was just shocked by the sight of me in my Christmas jumper. But I hadn't had to see that so much while I was doing this. So I want to check quickly. Do we have any questions in the Q&A or the chat? Two are answered.
I did answer a couple.
Brilliant. Some people have you've already answered them. Fantastic. Thank you, Luke. So Luke has already been answering. He's in the thing, in the Q&A. So Luke, you're a second year student here. Why did you pick coming to Kent?
There were a few reasons. One being the campus in general was just really nice and kind of like green, open areas. It just really kind of felt like a lovely campus to study at and work at. And then specifically for forensics was the fact we had a crime scene house, the fact that we had Chris Shepherd doing a whole ballistics module that I don't think any other university offers in forensics. So that was really kind of interesting for me, being kind of obsessed with, you know, guns and weapons and shooting, all that fun stuff. And I think a couple of lectures that we had on an open day, so such as this one that I vividly remember having in person, which was a bit different than online, but it was still thoroughly interesting. And then one that Bob Green gave, and it just kind of, it felt like a strong family. So that was kind of just a few reasons I picked Kent.
Yeah, I mean, we are a family. That's really important to us. I don't remember when I gave that talk, Luke, was I rocking the Christmas jumper as well?
No, you weren't rocking the Christmas jumper. I think you
didn't have that experience. Yeah, you see. So the family thing is an important thing for us, and I think it's kind of a key part of what we like to do. We like to work and get to know our students very well. We like to go and do the kind of activities outside and take them down to experience the real world when we're allowed to. Obviously, not at the moment, that's much harder, but actually be able to do those and study that. I saw there are a couple of questions in the chat which you've already answered, but for the sake of this, I'm also going to ask them out loud to you, Luke, so you can answer again. Okay. Kind of how many hours of teaching do you get for a fortnight? What's the split between lab practicals and other lectures and things like that?
So at the moment, I think we're averaging about 16 hours a week, and we've got we're having labs being about 8 of those. So we've had 4 hours actually in the lab doing forensic techniques. We've got just actually come back from a lab practical in the woods today. So kind of full of evidence bags and wearing the whole noddy suit, which is just behind me, and all that sort of stuff. And then we have stuff.
You didn't fancy wearing the whole suit for this? You didn't fancy wearing the whole suit for this?
It's not the most comfortable, I'm not going to lie. And it was quite cold as well. So no. And then we've got stats labs as well, which unfortunately aren't online, which are online, sorry, not in the lab, but we had those as well. So they take about 4 hours each between that. So we did actually go in for practical work.
So we've had a question, a new question. Thank you. Please keep bringing those in. I know we've hit the hour, but we're happy to stay open and answer any of your questions that you want to ask. And one of the questions was, are we still expecting to get these hours? I mean, hopefully, with all the vaccines and everything, by this time next year, where the kind of issue of COVID-19 will be in our rearview mirror, hopefully. But with that in mind, still, it will be it's interesting for people. Kind of, how has things been affected with COVID-19? Are you still getting your labs and lab hours?
Yeah. So, like I said, we had our forensics labs still in person. Everything was kind of done really well. We had goggles, lab coats, masks, gloves as standard for a lab, and we did have our own bubbles in the labs as well. That obviously was out on the floor, so it was probably safer than being out in public.
So I hope that answers your question. I mean, I hope it won't be a problem by September when you're coming. But what we have done this year is we spend a lot of time over the summer making the labs kind of more than safe. One of the advantages of forensics is the fact that actually a lot of the time you are kitted up and you are wearing protective equipment anyway, naturally, so it's safer. But we also had people working within bubbles and spaced out within the labs so that you were able to feel safe. Did you feel safe throughout, Luke, when you were doing it?
Yeah, definitely safe. There was one-way systems throughout the labs, and obviously those own little bubbles that no one else would walk in. So yeah, really safe.
Fantastic. Another question that we have in here is, what are your average class sizes for this course?
So we have 65, I believe, in our year, and that's quite big for a forensics class, I've heard. And we do share some modules with chemistry students as well. So we average probably about 100 if we're doing a chemistry and forensics module, or 65, like I said, for pure forensics modules. So it's small enough that you kind of, everyone gets to know each other and you feel like a good group and like a family.
We yeah, I think looking across the years, our course is growing in size, but it's what we try and do is we have a mixture of kind of small group and large group teaching across these things. So we have some large group lectures. You also have some small group teaching for particular things. When you're, for example, in the labs, you rotate around in small groups to a different thing on a particular week. So you still have that, and you have it sometime, for example, in the ballistics lab, you'll be able to do in even smaller groups and those kind of things. So that varies. What we have made sure, and actually we're expanding. So we've, as you saw, we brought in Andrew Langley as a new lecturer on the course, and we're expanding in order to make sure that we also support our students and make sure, because for us, kind of getting to know you and having that personal relationship to know everyone in the labs and in the lectures is an important thing for us. So we want to make sure that you all have the time and you have our time in order to see it. The last question we have in here is, I can only assume from Ollie Sylvester, who thanks me for a great talk as always. I assume this is actually a former student. He should be here. Who's a former? He's hijacked it, but I'll answer his question anyway about science. Who's turned up as well. Hi Ollie, if it is you. He says, thanks for the great talk. Regarding the use of drones in an instance scene, are there any considerations for downforce wind created by the drone's blade? Excellent question. So that is something that we've looked at. So you can, the drone that I showed, the one that I showed with a lot of the equipment on, is actually a larger end of the things, and actually probably isn't maneuverable enough to be able to take into most crime scenes. And that does create a downforce, which is a problem for disrupting things. We're actually looking at, and a lot of the work is looking at, more lightweight drones as an initial step. So your first step would actually to take a very lightweight drone in there, where downforce would not be a problem, because it's far less than even a gust of wind from opening a door, in reality, the downforce. And so there we would only put more lightweight sensors onto the drone, maybe even just an optical camera at that starting point. And incidentally, we're even actually starting to work with not only taking that imaging technology, but link it in with virtual reality. So a bit like some of you might know, things like drone racing and things like that, we're trying to use the same thing, so that actually a senior crime officer's able to go in and look around as if they would in real life, but the drone's camera is responding for that in exactly the same way. So using that kind of immersive technology. But you would start by sending in very lightweight kind of drone, and then once you've identified where there might be anything of importance, you can then send in your specialist drones with the specialist bits of equipment, which will be heavier, but by then you'll have been able to have mapped your scene by understanding on a basic level, in order to kind of identify where the important things are, bits of evidence may be, and identify where any hazards might be as well, because that's an important thing in the scene of crime, is not only identifying the hazards, but mitigating and making sure you're not contaminating. I hope that answers, Ollie, and it was nice to see you if it was you. The next question from another attendee we have, and I don't know, are you on the placement scheme or not, Luke?
I'm on the 4-year integrated master's course.
So you're on the 4-year integrated masters. So one question that we have had in the Q&A is, are there any placement opportunities? Yes, absolutely there are. In fact, one of the courses that we offer is a 4-year course, where the 3rd year you can spend in placement in a variety of different places. We place people in kind of the CELMARK and the places where a lot of so forensic science has been partially privatized in the UK as it is. So we don't place people into police forces, but we do place people into analytical labs. We place people into food industry doing a lot of work in the food industry. We have also we're also launching, which hopefully in times of COVID will return. We're also building partnerships so we can even have an international exchange placement year. We have had students, particularly students from those countries, who've actually gone overseas. So we had someone working last year who's just come back, who's working at The Hague, working on a fantastic project looking at DNA comparison, contamination, and detection of various things at The Hague. So yes, placement opportunities are there, as we've got a fantastic kind of range of people. So my experience working in the food industry, Bob and Andrew's experience is actually working in police forces. Donna Arnold, who I mentioned as our lecturers, is also, she's one of her interests is in kind of forensic heritage and heritage crime. So she has a lot of contacts into that world. And actually through any of these worlds, particularly in kind of the analytical labs, those things, we have opportunities for placements. And to go in and get you that experience in order to see it. And in fact, specifically, even more so, we have a placements officer. So we have an employability officer, remembering that one of the 3 E's, employability, is key to us. And we have an employability officer actually embedded in our division, Anna, who's brilliant. And not only does she give lectures throughout the course in order to talk to our students about what it's important, employability, thinking about those transferable skills and how they can be used, but she also has one-to-one meetings with every one of our students to actually say, well, what are you interested in? Where do you want to go? Helps them build their CV, helps them write those applications, helps them understand exactly what they want to do and where they want to go with that career in the future. So, and we have that actually embedded within our division in our school, so that you can think about that, not only for placements, but your career onwards. If you wanted a placement to be with the police, would that be possible? So placements with one of the limitations we have, and why we can't take a lot of placements to places like the police or even kind of more legal things, is because, and this will be a case, not just this will be a place for any placement within forensic science across the country, but possibly across the world, is that in putting a student into a placement, we potentially taking someone who, yes, you've been trained brilliantly, but you'll be your third year when you go to placement, and you're not qualified, you haven't gone through the qualifications. And there's a whole host of concerns from any employer that should you be processing a bit of evidence or doing something, and you at some point that evidence gets taken to court, if an opposition lawyer or that's challenged at all in the adversarial court system that we have in the UK, that could be challenged by saying, oh, but that student was just a student. It was just an undergraduate student who collected that bit of evidence or analysed it or did those things. So how do we know we trust it? And then and we know because we've trained you. They know because they've taken you on and they've trained you and they've probably got that backup. But the problem is that is an additional doubt that you don't want to do. It's a doubt in the juror's mind or potential magistrate's mind of anything that's going on. It's very hard to play into, and this is true anywhere to place into those line jobs, unfortunately. However, a lot of our students, what they do on the side throughout their courses is actually work as police officers, work as in a voluntary role in the police, particularly if that's a kind of career path that you're interested in. We obviously can support that. We have people who were former employees in the police, and we have a fantastic range. I'd say maybe 20%, 10% of our students every year go off and work in a variety of roles in the police, and once they've graduated. So we will support you through the process of going through those things in the voluntary roles in the police, so that when you graduate, you can go through their training into a final full entry into the police. And that is the last question that I have up here. I don't think I've missed anything. Thank you. Let me just check here. Oh, yeah. Okay. I've noticed in the chat that some there are, in fact, the YouTube we've got a variety of things on YouTube that are able to you can play. There is a playlist, so for any of you who weren't able because of internet problems to see my full talk, apologize for that. It is sadly the world we live in. It's just bad luck that tonight that happens to be the case. But we have a YouTube playlist. I am going to put that YouTube playlist in here for everyone, should you want to look at it, where we talk a lot more. There's some interviews with some of our current students in there, as well as some of the other things we talk about in forensics. So you can look and hear a bit more. Oh, one more question in Q&A, I see. Or did I imagine it? I now can't see it. Can you see the question in the Q&A? Ah, sorry.
It was just the one about the police.
Ah, sorry, perfect. So I hadn't done that, I answered it. Fantastic. In that case, thank you to everyone for coming. Please get in contact. You can find me on the website if you want to follow up and ask me any questions. My name is Rob Barker. So r.barker@kent.ac.uk. I'll put that in the chat as well. Okay. Please do get in touch. If you have any questions about the course, or if you have any questions about anything else that you would like to know, want to hear more about the research, and please do that. It's good to see you, and hopefully see you in Kent next year, and you'll see Luke as well in Kent next year.